That was a big deep dive into the next generation tools. We did want to make that the focus of this year's workshop because it is launched and it is very exciting and it has been long requested by our users. But now we'll change the tack a little bit, and we're going back to the analysis resource to look at the T-cell class 2 tools, which aren't available yet, as you saw in the next generation. So we'll look at it at our existing analysis resource. So that will include binding, processing, and immunogenicity. So with that, I'd like to reintroduce Bjoern again to present T-cell class 2 tools.
I will talk about class 2 now, and the tools are as Nina was just saying, and I think it doesn't hurt repeating, these are still the what we're now calling the legacy side of the IEDB Analysis Resource. So MHC class 2 represents epitopes to CD4 T cells. What is the difference between class 2 and class 1? So now garbage collectors, immune system, I think right after that. So the difference here, the other important difference for MHC class 2 molecules is that the cells that they are presenting this to are CD4 plus T cells, which are regulators, helper cells of the immune system. They provide help both for the cytotoxic CD8 T cells and also for B cells for antibody production and stimulating somatic hypermutations. So they have a key regulatory role to actually both activate and suppress immune functions. And in addition, and that has been more recently appreciated, they also have direct effector functions. There's cytotoxic CD4 T cells that can directly kill. Lots of them make cytokines that are really important directly without any kind of helper function to have a role. So this slide is similar to what Jay showed you for class 1. Class 2, the key difference is that there's two chain, the alpha and the beta, which both can be polymorphic. So that's the reason why when we talk about a DP molecule, we call HADP A1, 01, 03. This is like the alpha chain, and DPB1, 02, 01, because this is the alpha beta chain heterodimer. For DR, which is the best studied locus, actually, the alpha chain, the alpha chain is monomorphic. So there we typically just refer to the beta chain. So we say DRB1 or 101 because the DR alpha chain, there is just essentially one out there. The DRB345 locus, which is, to just confuse people, this is one locus, it's called 345, is in tight linkage with the DRB1 locus. And that's why often people don't type it. But bioinformaticians typically don't know which DRB345 allele goes with which DRB1 allele. So they should be considered, and they're just as important, like DRB1. So this is the kind of shape of the MHC1 of the MHC molecule. The other thing I should talk about is that how that then affects binding. So and actually that doesn't really, I was just talking about the heterogeneity, but the other thing that happens is the binding cleft, the way it's formed, allows the peptide to protrude on both ends from the binding core region. So you end up having peptides typically between 13 and 25 amino acids, which are binding through a core that really forms the contact to the MHC molecule, and it can hang over quite a bit. And identifying where the binding core is makes this more challenging than the class 1 prediction. So you really have to kind of pre-align the peptides to see that, say, these three peptides that look very different actually contain the same region here, and will bind most likely with the exact same affinity to an MHC class 2 molecule that likes this core. So as I said, these are the canonical, classical, legacy, or however we want to call it, IEDB analysis tool page, and here you can select your class 2 predictions, and then you get to this page. So this is the same page exists for class 1, similar to like the next gen tools. You have to start by entering the sequence that you care about, and then you have your prediction methods of choice. So for class 2, like for class 1, we have a recommended epitope prediction method, and that is based on these benchmarks that we're doing. This is the link to the class 2 benchmarks, which have found now for quite a while, similar to for class 1, that a net MHC2 pan 4.1 binding affinity prediction has by far the highest correlation with the measured binding affinities. And this degree of correlation has gone up massively, and you can't really see it here because these are like rankings. So this means it consistently performs high in the rankings, but also the quality over time has greatly improved compared to the methods that we had back in the day, like TepiTool. So there has been really a massive step up, and that is primarily due to the integration of ligand elution data for both training the BA and the EL method, which has given like a vast amount of additional data to train and has ranked up the performance. So if you want to predict binding affinities, you should choose the BA output. If you want to predict which peptides are naturally processed and presented, you should choose the EL output. The next thing that you choose in here, this is kind of the allele selection that we have in our legacy site. You first select the species and locus, and once you have that, you can then choose from a list of available alpha beta chains for that locus that you have selected. Note that you have the ability to select the 7 allele reference set or the full reference set as well. So this is just highlighting what you're selecting, and the 7 allele method. Okay, I haven't even introduced this yet. This gives you access to what we would currently recommend as the way to predict peptides in a broad population that are going to give you the largest response. And the way this was done was we took 6 data sets which were quite diverse, that measured the binding the T cell response in overlapping peptide sets, and for each of them measured the T cell magnitude of response, and then try to retroactively say, could we have picked these peptides, and limit out our ability to the synthesis of peptides to those that are giving a broad response in many individuals and a large T cell response. So, and that led to the selection of these 7 alleles, and you'll actually notice there's 4 of the 7 are from the DIB 345 locus, and the reason actually for that is that because that locus is more frequently found. If you cover these, most likely your peptides are going to bind at least one person, because pretty much everybody has one of these alleles. It's not pretty much anybody, I should be saying this, but they are quite similar. So the selection of these 7 alleles are essentially representative of common binding specificities found in the human population, and then by using that set, you'll get predictions for these 7. You can then choose the length of peptides that you want to make predictions for, and then you get predictions output that has the allele listed, the sequences coming from start and end. You see many of these things are very similar to what Jay showed before, and you don't have the same kind of filtering options and stuff like that here, but at least you can sort, and you're getting here the sequence back and the output sorted high to low by the score. But so here, for example, actually the only thing, okay, this is not even 7 allele. Never mind, I'll talk about that later. So how do you select peptides? So previously, we had actually through every year until this one, we have always said MHC2 predictions are much worse than MHC1. So you have to be much more careful in selecting them and be more generous and allow for more tolerance essentially for predicted lower affinity binders. But the difference, as I said, the quality of the MHC2 binding predictions has massively improved. So now we would loosen that or justify to use stricter cutoffs, but we haven't updated yet. We haven't yet systematically analyzed how does this translate? What is the new cutoff? So we would still now stick to cutoffs we had set before. I mean, this is what we've been using, is like a 10 percentiles for class 2 binding or MHC binding affinities of 1,000 nanomolar or less. This is both quite a bit looser than what we would recommend for class 1. And I would now say that part of this looseness was based on that these predictions just weren't as good. And now we can probably be tighter and go to ranks of, I don't know, 5%, 2% or less. But we'll really need to do a thorough evaluation on T-cell epitope, on the effect on T-cell epitopes that comes from this. And that's kind of what I put here in bold. So this might be, you can consider this like a preview of what's going to be coming. We are going to reevaluate what we have as recommended thresholds for class 2 in the upcoming year or so. So the way this is working, it does all the overlapping 15 mers and that are in the input sequence, and you can see here that essentially you have DHLE, HLE, etc. So you have the peptides that are essentially all of these are predicted as good binders, but they are really the same peptide because they are kind of overlapping the same region, and they have the same core that is actually being identified as binding to class 2. So to get rid of that, one approach is post-processing. You just do the whole predictions, and then you go in top to bottom and remove peptides that are close to the current start positions using your favorite script or so. So as I showed you, this is just one example of a number of potential post-processing mechanisms that you might want to do with your epitope predictions. And that's actually one of the reasons why we did the whole pipeline approach and the whole combine multiple predictors into one that Jay was showing you for class 1. So we don't have that for class 2 yet, but actually what we did for both class 1 and class 2 is to implement some of the common steps into TepiTool. So TepiTool was our previous approach to pipelining, and for class 2 we very much recommend that because it does several of these steps that you would otherwise have to do manually, either by pre-processing or post-processing your predictions to get to the final results. So I'll show you that TepiTool pipeline, which again works for both class 1 and class 2, and it's quite similar. I'm going to go through, there's multiple steps to this pipeline, and I'm going to go through these steps, and then as I do, at the end, point out where there's going to be differences between class 1 and class 2. So the sequence input is the first step for both, and it's as before. You can type something in or upload a file, and then you go to the next step, which is going to be your allele selection, where you have again, like you select the species first, so this is limiting the alleles, and then you have the specific allele selected. And as Jay was pointing out before, it's like these controls aren't there on the next gen side, but the same alleles are there, and you just have to know which one's which. So this then echoes what you currently have selected. You have your 3 sequences. You've selected human, and then you can ask, do you want to do class 1 or class 2? So this is the bifurcation then in the tool afterwards. If you want to do class 1 selections, class 1 alleles, you're going to get to this page, which gives you the lists of frequently occurring alleles. That's what's here. We should have pointed this out. I think we've said this before, and somebody asked a question about, can I make predictions for all alleles? All alleles is something like 5,000 or even much more. There's typically no, unless you're really sure, there's no reason to do predictions for 5,000 alleles, most of which are going to be very similar to each other, and most of which are going to be found in less than 0.00001% of the population. So this is focusing on ones that are at least at a 1% population frequency. So some of these are still quite rare, but at least there's some evidence for them. You have other choices here. So like you could do our panel of 27 that Jay was using, for example, or in this case, we just select 3 alleles, again, like on the right-hand side. It just summarizes what you currently have selected. Then you have you at the peptide selection things. You can have like default settings. Do you want to have a low number of peptides or a high number of peptides? How do you want to handle duplicates? And this is actually then preset. If you set a default, you want to have a low number of peptides, then duplicates will be removed, and only length 9 is recommended for class 1, etc. So you have these different settings here. Then you can go specifically to the length settings, like, okay, when you do custom selection, you can now do all of these by hand. You can remove duplicates or not. You can say specifically which lengths are you interested in. And you can also choose this conservancy analysis, where you're asking, as an additional step, I want to know that all the peptides I'm selecting are also conserved in 50% of the sequences that I had as an input. This is if you go back to the dengue example that we had, so this would be saying you only want to capture, you only want to deal with peptides that are found in at least 2 of your input sequences. So the method selection for class 1 then allows you to switch which prediction method is used. We have the same methods that we saw for class 1 in Jay's version are available here. You have the IEDB recommended, etc. And you can, after the method selection, identify. So how are you going to choose your threshold? Do you want to do peptides based on the predicted percentile rank, predicted IC50, based on the MSC specific binding thresholds that Jay mentioned in TepiTool? They're directly implemented, so you don't have to do a table lookup later yourself. You can just use these thresholds that are pre-stored here, or select the top percent of peptides or number of peptides. So for all of these things, you can find help on the threshold selection in the IEDB Zendesk system, and pointing to specific papers that that justify these thresholds that we've been using in the past. And again, those are going to be very soon updated. Then you can again. So what is this one? Sorry. So these are the alleles that are being used, and there might be a correction if you choose a certain prediction method, and it only has a subset of the alleles available. It'll list those. I mean, in general, this only happens once you go away from the recommendations that we would have for which prediction method to choose. So you have your 3 sequences. Human class 1, you have the 3 alleles that were chosen. You're in this the way we've chosen this now. You're removing duplicates peptides, doing 9 and 10 mers, and then this means that you have 114 peptides that are kind of going into your predictions, and then you're going to filter out by conservancy. Then you get to the final page where you can submit these this prediction task to you. You can enter an email and a job name to this if you want to run this. And again, this is for if it's going to take a long time to run. If it's fast, you can just wait for the page to refresh and be done, and just submit. Now I want to highlight the differences for class 2. For class 2, we have again, we have default settings for certain things that we then recommend. So for example, okay, this actually summarizes for low, moderate, and high number of peptides that you want. What is then the approach in terms of how many overlapping residues are allowed, how many duplicates allowed, yes or no, and then it gives you an estimate of the approximate number of peptides that are going to be coming out of this with the current settings. So again, you can select these. And I think I didn't say this really before, like why do we go with low and moderate and high number of peptides? In reality, when you're thinking about running experiments, very often the reason for or what is driving the number of peptides that you want are sample scarcity. You will not be able to say if you have blood from a sick individual, you're lucky to test maybe 20 peptides or so in that individual. And if you have like a large pathogen to start off with, you should totally go for a low number of peptides. Versus in other cases, maybe you can get apheresis from a healthy individual and you want to test, you can test tens of thousands of peptides. So that's the sample availability. And the other one is money. Every peptide that you synthesize is going to cost. So the number of peptides is actually very often driving and obviously the experimental effort that it takes to then screen these. So obviously the lowest number is the lowest number of peptides then with the highest confidence that they are real hits. And with a high number of peptides, you're going to make sure or are going to be more sure that you're not going to miss any predictions. So that applies for both class 1 and class 2. Now again, you can run this and get your summary. For class 2, for the allele selection, I should point out, it's the same thing with the allele selection and sorry, with the review and submit, you're getting back to what your the summary of the job that you have entered. So actually, this is the one I was looking for. There is an alternative method for class 2 that is not present in class 1, which is here. You can select peptides based on predicted number of alleles binding. That is exclusive to class 2. This is because for class 2, because of how I showed you, you can have a different register in which a peptide can bind to MHC class 2 molecules. You actually have quite frequently peptides that bind different alleles that might actually have very different binding specificity just because the alleles can bind in different offsets. That really doesn't happen for class 1, so there that option doesn't make any sense. So you will have that only here, and then you can do things like predicting peptides that bind at least 50% of the alleles that you're interested in, which does make sense for class 2. You can also for class 2 select directly the 7 allele method that implements what's in the paper, what I showed you before. Again, this not just then selects the 7 alleles, but also does the selection like we've done it in the past, namely calculating the median affinity of this over the 7 alleles as a predictor of promiscuous binding. So to summarize the MHC2 binding predictions, I showed you that, binding for class 1 molecules, binding for class 2 molecules, and TepiTool, which has the different steps in this pipeline implemented. And there's different things in here, namely the processing predictions, similar to what we have for class 1. And here I want to talk about MHC2NP. This is the one that we have for class 2. The other ones here are for class 1. And what this tool does, it actually tries to identify here these flanking residues that are the cleavage sites of the proteases that are essentially generating the MHC class 2 ligand. It tries to predict essentially where this cleavage happens. Now, I want to point out here something that actually took us a while to understand, that while these cleavage sites exist and we can predict them, as I'll show you, they typically do not really impact the core of the peptide that is in contact with the MHC molecule. And that's important to remember as I go through this. So we did essentially take a large number of peptides that we found from MHC class 2 processing data sets. And based on those, we looked for kind of the motif at the C and the N terminus of the peptides that distinguishes peptides that are directly eluded from a cell from random peptides that had similar other properties. And we could use these and clearly saw like a cleavage motif that allows us to improve the predictions of these ligands. However, what frustrated us, but really turned out true, and actually by several other people who had similar things, this does not help in the T-cell epitope prediction. I'm going to go through the tool here. So you enter your sequence, you click submit. You have then these peptide results, and it looks for at the peptide start and peptide end. You have this see as the S is preceding, then you have the VP, or you have the PKA. So it's always like the it covers the cleavage site that liberates this final peptide of interest, and then we can calculate a cleavage score probability. And actually, what's common to both ends is like these prolines are often signals for where cleavage happens. Prolines typically are not directly at the cleavage site, but are one past essentially, and they tend to like anchor these proteases at this position. So this is like a really nicely cleavable peptide. So we can export these results, including these scores, and we can combine them with MHC binding predictions. And when we then look for the ability to predict ligands, so again, like peptides naturally processed and presented, we see this improvement in prediction performance when we combine the binding score and the cleavage score, and it gets really nicely better than binding alone or cleavage alone. However, when we apply the same approach and try to predict T-cell epitopes, this totally isn't the case. And that again is because the and these were very completely separate studies, and we reached the same results, that we couldn't improve T-cell epitope predictions. And that's primarily because, as I showed you, the core of the peptides, which is also where the TCR binds, is pretty much not affected by the cleavage. And these motifs that we have are pretty common. So as there will always be a cleavage site within like 5 residues or so of the ends, and all of these peptides bind just fine in the same way. So essentially, you don't really learn much by knowing the exact ends, in contrast to class 1, where processing has a much bigger impact. So that was the processing predictions. Then we also had an attempt here on the immunogenicity predictions. That was done as follows. We extracted data sets of proteins from the IEDB, where there was overlapping peptides being used, and tested them for T-cell immunogenicity. We used these to train a neural network to learn essentially motifs associated with immunogenicity for class 2, and then had a score that specifically just asked like immunogenic or not, and combined them with the 7 allele binding score, which is like binding or not, and in order to improve these prediction performances. We had an in-house data set. We used a tetramer control data set from the IEDB. We used 57 different studies from groups around the world and implemented as I was just saying. And then this is essentially what you can do here. You can go in with your peptide sequence. You can use the combined method. That's what we recommend. Or the 7 allele method is just the binding or the immunogenicity by itself. Run these similar to before in the other tools, submit them, and then you get your combined score back, the 7 allele method and the immunogenicity score and the combined score. Now, the issue is that when we looked at this performance of this combined score, we are really just going from 0.704 or something up until to 0.725 or something, which, I mean, a change in AUC of 0.02 is great when you're starting at 0.95, but in this range, it's really not a lot. And so we have done additional tests on this method in the past years and have not found that it really improved immunogenicity predictions. So while at the start we were very happy with this, we at this point would not strongly recommend relying on this tool. That's also why it never lost the lab icon. We have not been able to consistently validate that this is better than just using the 7 allele method by itself or direct MHC binding predictions. So to summarize class 2, as I said, the MHC2 binding performance and ligand dilution prediction performance has massively improved in the last years. And actually, some of the last graphs I showed you were still about like the traditional MHC binding predictions. These would have gone up quite a bit. So the thresholds that we've been using, this is for allele-specific predictions, have not yet been updated to reflect that. And so we currently would say still top 10%. I mean, that's not what we still have in our documentation, but internally we've been using more stringent cutoffs with success. We would recommend the net MHC2 pan 4.1 BA scores to predict when you really care about the binding affinity. If you care about ligand dilution, we would use the EL scores and also for epitope predictions. Now note that those actually, they'll give you, if you want 50 mirs, they'll give you 50 mirs. They don't care about the ends. They care about the core essentially that is binding to the MHC molecule. The promiscuous nature of MHC2 binding makes the predictions for class 2 different than class 1. There's 2 levels of promiscuity. One is that multiple class 2 alleles actually have very similar binding motifs. And then secondly, the same peptide might be bound in different registers by different MHC molecules. And that overall allows you to identify protein stretches or longer peptides that might be commonly reactive, broadly reactive. And that's something we found extremely useful when you want to design diagnostics or just reagents to pick up antigen-specific T cells, because they all work in a broad populations, and you don't have to like a customized peptide selection for based on the HLA type of the person. TepiTool is implementing the pre and post processing steps that we have for T cell epitope prediction for both class 1 and class 2. Now for class 1, we would recommend at this point going instead to the next generation tools, and there, as we are adding to our pipeline capabilities, essentially there'll be just a link in which you'll click on and say, this is now the recommended IEDB class 1 epitope prediction approach with minimal low number of peptides, medium and high number. We don't have that yet done, right? But you can see the vision that we are trying to have these things as essentially a link you can click on and say, this is how I made my predictions. And that's also another thing I wanted to point out for this pipelining and ability to store a pipeline will make this sharing easier. We've had people who said, I'm using the IEDB tools and I want to cite in my paper how I did this. How do I describe it? And there's a lot of like, I use this and that and this and that, and it's really hard for somebody else to really know for sure that they did the same thing. If we have the whole pipeline with one link in there and that's persisted, that's going to facilitate sharing. It's also going to make it easier for us to say, we used to say choose top 10%. Now we would say top 5%. And then we can document that and say, this is our new recommendation. And this is the whatever version December 2023, which is different from the version of June 2023 or so. Okay, that was a bit of a digression here, but I'm trying to motivate like these kinds of changes are easier for us to communicate and to share by having them as multiple pipelines compared to the TepiTool approach, which is kind of like, that was our first step from, we have each tool by itself. In TepiTool, we tried to change it together, but we had like one kind of, it was still very guided, and changing that became really hard. So we are hoping in this sharing of pipelines, it's going to become easier. And I guess all of that was what I was trying to say here. The next generation tools will implement this TepiTool functionality with more flexibility and ease of customization for whatever your use case is. And I always have to say this, all of our recommendations are based on our current best knowledge, and this is always being reevaluated, and this is science. This is active science going on, so it would be no surprise at all if a year from now, next year, there would be changes coming, and the year after again. as we learn more and gain more experience. I think that was my last slide. So I'll stop here. And I believe that was pretty good.
Thank you so much, Bjorn. Yeah, we're running about 5 minutes ahead of schedule, so I'm going to give you a minute to catch your breath and also take a look at the Q&A. We've got...
guess what happened? The fun thing, I lost my questions. Because of the blackout, I only see 7 questions.
Of course.
Is somebody at the institute and has the stuff here and can come up with a laptop, or I can run there? I guess, I don't know.
Everyone else is at home. What I can do, Bjorn, is the ones that you've indicated that you will respond to, I can read out on your behalf. Yep, these are the fun things of virtual live events, everyone. So thanks for your patience.
Somebody can tell Zoom that it's not a good idea to
And that gives us our official answer. So if you drop off, you do lose the history of your Q&A and chat. So anyway, while I let you maybe take a look at the new questions, I will like to invite Jay back to the floor. And as I mentioned, we've got quite a few questions, a couple of extra minutes, so we'll do our best to answer them. So Jay, did you want to tackle the first question?
Sure. Excuse me. Yes. So the first question that I see for me is about about the alleles that are available from on the NextGen site. And so there's a question, are we limited to HLA only? And this is from Sham 28. Or do you have to select the individual alleles manually? And I just want to share my screen again to kind of try to convey how that's working right now. So I'm going to go ahead and do that. Right, so we're on the NextGen tools page now. And how we have the allele selections working is just as an autocomplete. So you can type your favorite HLA alleles here, and it will autocomplete to the best of its ability. But you can type, like, for instance, mouse allele like H2, and you do have to enter them only one at a time at this point through this interface. You could enter them one at a time, you can remove them one at a time. The only functionality that we have right now in terms of kind of bulk adding them is through this this pop-up window, which is this finder where we can add that 27 allele panel in bulk. We are working on enhancing that functionality and making this finder kind of an actual finder with a tree view, similar to what you would find on the IEDB website, where you can be able to select multiple alleles and in a single go.
Awesome. Thanks, Jay. Then
okay, go on.
You want me to start earlier?
Sorry?
Sorry, would you like me to start with one of your earlier questions, or do you want to take most recent one?
I do the ones that I have open now.
Awesome.
Okay, Maricanta Bondada, while selecting alleles for MHC class 2, which locus we should prefer, DP, DQ, DR, or we need to select all the locus and select best binding epitopes among them, which will give best results. So the way the predictions work, so first of all, the 3 loci, it'll depend on your specific questions, but in general, HLA DR is typically higher expressed than DQ and DP. That's why it's more frequently studied. But that doesn't mean DQ and DP aren't important. That might depend on your question, but DR generally has some advantages. You should be able to choose one locus, choose one allele, choose the next locus, choose the next allele, and then do them all one after the other. Ultimately, when you compare them, you should pick whatever you care for. Do you care for promiscuous binding across alleles? Do you care for a single best allele for peptide for an allele? It'll really depend on your question. Jay?
Sure, yeah. So the next question for me is from Flavio. Can we send many REST requisitions in parallel which is the maximum? Can we send many or all alleles requests and many peptide things? So there's a few questions there. In terms of alleles and peptide lengths, yes, you can request many alleles and peptide lengths all inside one request. You don't need to do a parallel request to do that. That said, you also can send parallel requests. Currently, we do not have any rate limit set there. I'm sure we're going to work on establishing that because we can't have rogue users bringing down the servers. So we'll need to establish some guidelines there, like we have on the legacy site.
Awesome, thanks, Jay. Next question I have is Laura Hartman. Do MHC class 2 predictions methods consider the flanking regions, or is prediction only based on the binding core? So there's two answers to that. The class 2 binding predictions, ultimately the way they work now, they do take the flanking regions into consideration, but the weight essentially, and this is done internal in the neural networks that we would recommend, they internally train on the variable importance. And so when I talk about it only takes the binding core, I'm really simplifying. It does take the flanking regions into account. Jay?
Yeah. All right. Next question is from Anonymous, and it says, how long is the data associated with these shareable links cached? So our goal is to be able to cache them for a very long time. And so that's kind of a non-answer, but yeah, the goal is to be able to provide you with these links that you can actually use in publications and hopefully share them indefinitely. And we're working on a mechanism to do that. So currently they're shared, they persist indefinitely.
My next was from Paola Flores. Sorry, how I perform the best post-processing by removing the redundant peptides after HLA binding predictions is by using TepiTool. So there's the two ways. I think the best approach is to just use TepiTool, which does it for you. The other approach, I was really just trying to point out how annoying it is, would be that you export the results and then write a script or so that for yourselves does this removal, or manually going through. So, but it's much easier to do this in TepiTool. And ultimately, in the next gen tools, we will also include similar kind of functionality, like removing overlapping peptides, etc., directly in the next gen pipelines. Jay?
Yep. So next question that I have is from Hur Al-Hassani. It says, are you planning on making the toolkit available via Conda? And I think this question was asked yesterday too. I'm not sure if it was from the same user. Answer is maybe. So we don't have currently a plan to use any kind of package management tool right now. Just as we've done for the legacy site, we kind of package up our own tools and distribute them as tarred zip files that you can run on your own hardware. And we give you instructions on how to set up your environment for the tool. But that definitely is a possible path that we'll consider.
My next one is from an anonymous attendee. Hi, can we check in the prediction results if the peptides have been experimentally tested already? For example, MHC binding assays with either positive or negative results. That's actually an excellent suggestion. We are currently not doing that. It's something we've talked about, and that would be actually an example for another piping tool where the output predictions are essentially run as queries against the IEDB, and the data is being retrieved by that as part of a pipeline step. But yeah, no, so you're thinking ahead, and we don't have that yet. Jay? I can also do more if you want. I'm picking easy ones right now. Are the 7 alleles from Emma Ryan? Are the 7 alleles selected from global populations? When you look at their frequency, they are all frequent in global populations, and yes, they weren't selected as such. They were selected based on their ability to predict T-cell recognition in a quite diverse number of different populations. So implicitly, that led to the selection of alleles that are frequently found. but also not just based on those allele frequencies, but also that their binding specificity seem to be reflected probably in multiple alleles that are just very similar, that add up to an overall good global coverage. Jay?
Yeah, so I have another question from Flavio that I'm going to want to share my screen for. And it says, as you showed, is it possible to build a pipeline and reuse it? And also get the JSON and run locally. So let me quickly share my screen again. And so now we're at the, sorry, there's a lot of Zoom stuff in the way here. We're at the T-cell class 1 page. And I mean, we just ran this one tool. Everything, we call everything a pipeline, even if it's just one step. So pretend there are more steps here. Yes, to reuse the pipeline, you just go to this menu here. You get a link to this pipeline. You can copy this. You can open it in another browser window, and it will load up with the data attached. So yes, you can reuse it. In terms of running it locally, if you have the standalone, you can, well, I guess one more thing here is you can also copy that API post request and bring it to the API documentation where you can fill it in and execute. I guess that's not locally, but to run it locally, you would download this file here, which is download the prediction parameters, which is a JSON format file. And open that up and bring it in to show you what that looks like. And that is a file that is compatible with the format for our standalone tool, right? So it tells you now we've selected these two alleles. We want to predict for length range 9 to 9, and we want to run an NMHC-PAN-EL binding predictor. So you can, once you run the pipeline in the web interface, you could download the parameters and use those on the command line as well. Bjoern?
Thanks. Next question is from Hatim, Mutivala, and Zene. And apologies, I'm probably butchering names left and right. The question is, does the tool predict immunogenicity of peptides with modified amino acid? For example, can it predict oxidized methionine, tyrosine, and peptide sequence? And there's a follow-up. I mean, can it predict immunogenicity of peptide with oxidized amino acid? And the quick answer is no. I think Alex answered this yesterday as well. Ultimately, there's not enough data that we have in hand on the immunogenicity of these modified amino acid sequences. There have been some efforts to specifically look for phosphorylation and for citrullination. And there are tools there currently being still developed that might make it at some point onto the website. Right now, we have nothing. Jay?
Yeah, so the next question, I accidentally just clicked it away, but it was from an anonymous user that was asking if we, for the immunogenicity prediction, do we take the position of the amino acid into account? And the answer is kind of, but not really. So we don't really take the position into account. The only position that matters is if it's a masked position. And so there's a selection. When you run the tool, do you want to mask the anchor positions? And that means that those positions are just going to be removed from the calculation. All of the other positions are weighted equally. Bjoern?
Thanks. Next one is Manikanta Bondada. When we predict T-cell epitopes in IEDB Analysis Resource, will it automatically follow all steps in TepiTope in predicting epitopes, or we need to use TepiTope separately? You will need to use it separately. If you just follow the default setting and just click next, 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 there's going to be a few clicks, and then you do the standard workflow. But as I showed, in every step you can modify it, and that's the only way to run TepiTope and yeah, to implement these. Jay, and that was my last one of the ones that I had clicked yes to. Jay, I'll afterwards now would go to things that Nina sent me from previous ones selectively, and then Nina, if you can click yes after I answer something, but go for it, Jay.
Sure. Okay. And I'm going to want to share my screen again for this one. So this is from Noor Al-Hassani again. So the API works so far for tools and queries. Is it possible to access and retrieve the data as well? If yes, do you have some examples in the query? And I'm not 100% clear on which API we're talking about here. Am I sharing the right screen? Does it show? No, it's the
It
wrong
shows a
screen.
lot.
Yeah, that's the wrong screen. I'm sorry. Okay, that's probably better. So as I just showed you, I ran this this prediction, and we have a we copied that post request. We could run that through the API. When we do that, when we do this, this actually is executing live. We get a response back from the server, and oops, and we're going to get the results URI. And that is where the data will be. So the result data, once it actually completes running, and it looks like it's complete. So it's in this JSON structure inside CBC status done for this one. Inside data, we'll have a bunch of stuff. So we have the peptide table that includes the data, and then a description of each of the columns. Then we have the NetImageCPAN allele distance table and the input sequence table. So that's how you would get the data back. If you were referring to the API for the database site, we're going to talk about that tomorrow morning. So I'll show you some fun examples there.
Okay, I'll take the first, I guess the second one from Omar Sheikh. Since the peptide binding cleft can only accommodate 8 to 11 amino acids, this is for class 1, what's the advantage of having peptide slider options in the IEDB tools to more than 11 amino acids? So the binding cleft can bind more. There are well-documented and perfectly crystal structures and immunogenic epitopes up till 15 and potentially even more. The thing is, they're extremely rare. So typically it is not, unless you really, really care about longer peptides, 11 is a practical cutoff. And then you're going to cover 99.5% or so of all peptides, but it's possible to go for more. So we allow for larger ones, but we would not recommend doing it unless you really know what you're doing.
All right. And so I see one from an anonymous user. What is the guideline for choosing a threshold value for rank, calling it significant, like 1%, 5%, or 10%? And so as I was trying to convey, so we have a few kind of general guidelines, and it depends on which tool we're talking about. For class 1, we generally recommend 1% as percentile rank as the cutoff, and which has been shown to capture, I don't remember the number, but something like 75% of binders for that peptide. For class 2, actually I don't remember the current recommendation. I think Bjoern is probably more familiar with that. But it's higher than for class 1 because the performance of the class 2 tools is not as close, as good as class 1.
I'll take the next from Tarun Jarai Navani, Lonza. Is it implemented or can it be implemented in the pipeline to use the potential peptides from NetShop directly to predict class 1 or 2 predictions? Actually, so you saw, we can do all these predictions side by side, and then you can combine filters and say you can select that based on the NetShop predictions being above a certain threshold, filter on those, and in parallel then have the class 1 or class 2 binding predictions in there in the same columns, and that would be the way to implement that. I'll just do the next one. Or Al-Hansi, in context of finding similar epitopes to my target, is it possible for the HLA allele to bind to similar epitope with different length? Yes, that is actually quite possible. That is actually quite common, that you can have an extension typically at the N terminus, by a residue or two that still binds. It depends on the allele. Some alleles tolerate this, others don't. So the best approach is just trying predictions with different lengths. And then I'll do the next one. And Jay, you can do the answer live thingy also on the new ones. I didn't see any there yet. I don't know if I'm not seeing them or I don't know. Anyway, Chris Thorpe, these are the ones I remembered before, asks, do you have plan to have any parts of the processing tool account for different N-terminal trimming by ERAP alleles? We would be excited if somebody has a method that predicts this quantitatively. This is one example of, there's several others like Tepiton loading, and so there's a number of steps that are clearly important for the MHC class 1 processing presentation pathway, but the ability to quantitatively predict isn't as good as just using the whole ligand elution ligandome and kind of letting the neural network figure out what the relative importance of these steps are. But if that such a method was publicly available and we could re-implement it, we would jump at the opportunity. Jay?
Yeah, so I'll take a question from Carl Hanf. It says, so does the callus MHC1 immunogenicity prediction assume MHC1 binding? That is, does it give the chance of immunogenicity given that it binds to MHC1, not binding plus downstream process equals immunogenicity? And actually, that's a good question. So yeah, I mean, it was trained on a data set of known immunogenic peptides. So inherently, these peptides, I mean, they had to bind MHC. But that said, it does not take into account, for the predictions that it makes, it does not take into account whether or not that specific peptide has any kind of MHC binding motif in it. It just takes into account the amino acid frequencies.
Okay, so I'll take a different one. Anonymous attendee asks, what is the difference between PepMatch and BLAST? They are both very similar, and the difference is that BLAST actually doesn't guarantee you that you'll find a match. BLAST does a statistical alignment, and it relies on this E value to have like more than likely matches. PepMatch guarantees you matches. BLAST was designed for aligning longer regions, and PepMatch kind of fills a gap of finding exact matches or like guaranteeing to find the best match in a large sequence in like sequence spaces between 8 and 15 residues or so, versus BLAST works best when you have like 40 residues or something like that as a minimum.
Yeah, and so there's a question from Jing Du. It says, for immunogenicity score, what is the top and bottom score, such as for known antigen and known non-antigen? And so I don't know exactly, but the range is something like minus 0.7 to plus 0.7. So it's somewhere around there, with 0 being our cutoff for what we would consider potentially immunogenic.
The next one I'll take is from Omar Shaikh. What is the cutoff for PepMatch score for a potential autoimmune peptide? That is a good research question. We would think, based on other data that we have, that 3 or less mismatches in a 15-mer is probably a threshold for class 2. And that's just based on the general ability to do cross-reactivity for T cells, where we have some data from other studies that have been evaluating that. Now, I would not take that too serious, not like as a real massive cutoff, because it's just not well enough studied. Sorry. So yeah, it's our best guess.
So I see a question from anonymous attendee. Do you have any plans to speed up the MHC2 predictions? Currently, it's a little bit slow, especially when you have a large list of proteins. And so, yes, so when we get to migrating the class 2 predictions over to the next gen tools site, performance is one of the things that we're going to look at. But that that'll probably be around June of 2024.
Just a quick time check for you, Bjoern and Jay. We're at 10:20, which is the break time. Do you want to take another couple of questions?
So the break is starting now, or
Yes.
it started already? Okay. So I'm running out of steam, but let's do a few more. Okay, Laura Hartman asks, you mentioned that the MHC class 2 immunogenicity prediction is at the moment not better than class 2 binding predictions. Do you see the potential that class 2 immunogenicity predictions, I love that word, it gets better with time, will improve in the future? Absolutely. We do think that it should be possible to predict that, and be hoping with large, I mean, as always, more data sets, larger data sets are going to make it possible. Yeah, but I can't say when. I see the potential, absolutely. Can I tell you when we would have that? Absolutely not.
And so I see a question from Carl Humph. Is TepiTool updated under the hood to use your recommended NMHC 2PAN 4.1 BA? And the same question for CD4 Episcore. And actually, yes. TepiTool has actually been recently updated to use the recommended NMHC 2PAN 4.1 EL. When you select IEDB recommended, since that's the default IEDB recommended. For CD4 Episcore, I need to double check. I'm not sure. I'll get back to you on that later.
So, and I think, Nina, I would vote this is the last question I'm going to answer. Anonymous attendee, is the binding core predicted or unique? The binding core is essentially by taking the longer peptides and looking for the best matching 9-mer stretch within it, and that's given us the binding core. And if that best core is not good, it would still put it out. It'll always give you the best guess. So it's definitely predicted. And the fact that there is something in binding core listed, again, like it's the best guess, but it itself might not be a good binding peptide. You always have to cross-check, is the overall predicted affinity high? And only then does the binding core really make sense. And there might be 2 or 3 quite similar possible cores in the same peptide, and the tool doesn't try to make an attempt to identify that. And yeah, at this point I'm dead.
Okay, yeah, I can answer the last question from anonymous attendee. Maybe off topic, we're developing a tool for peptide MHC prediction. Is there any way to be included in the IEDB weekly benchmark? And yes, we have some instructions on the benchmark page on how to register your server to be included. There are a few requirements there, and I'll put the link in the chat also.
And we would love that. Yeah. So we want more participation. The problem has typically been, unfortunately, that many of these papers are written, the server is put up, and then it's no longer maintained. So the reason for the lack of external tools is often that most of those are no longer available in the form that we can run them in an external benchmark. Yeah, and we're trying to make it as simple as possible, and we'll work with you to make it happen.
Awesome. So with that, I would like to say a huge thank you to Jay and Bjoern. That was a really big mammoth block 1, section 1 of the day 2 workshop. So thank you both. Thank you for answering all those questions. And importantly, thank you to our attendees as well for bearing with us while, we had a few technical issues as well. But we basically answered all the questions. If there's anything else, we might type a couple of responses. But as always, if you have any follow-ups or maybe we didn't get to it, you can always email us at help@iedb.org, and I'll pop that in the chat as well. So now we are in our break. It'll be about 10 minutes instead of the 15 minutes. I'll pop the time in the chat for everyone to see as well as a couple of additional resources. So yeah, stretch your legs, have a little break, and we'll see you in about 10 minutes. I'll update the chat shortly. Thanks, everyone.